Now, if you've gone to any bioinformatics seminar over the years, you'll see a couple of different types of graphs. One of them is the growth in sequence information over the years, and everyone's familiar with that graphic now. But there's another graphic that we've presented in the past that describes how many resources are available over the years as well. So these are software tools that researchers need to access that either store information or provide mechanisms for you to analyze information. And we'll talk about the number of those overall in a minute, but here's just one snapshot of one particular site. So many of you will be familiar with the National Library of Medicine in the U.S. What they do is they provide access to a number of different types of resources that you might need. And so if you take a look at the graph that's shown here that they provided in their congressional budget justification, you'll see that they talk about BLAST being available by 1990, Entree, OMIM, 1996, PubMed, of course, many people will be familiar with PubMed, and there's many other tools that have been added over the years as well. So this is just one particular site, one particular group that's providing all kinds of new tools over the years as they try to manage the number of base pairs of data that are coming in and many other types of data as well. And as you can see, increasingly, over the course of years, users have to become more and more familiar with accessing these tools. And at Openhelix, we're lucky because we are not tied to one of these particular projects. We get to talk about all of them. And that's one of the benefits of learning through Openhelix. We get to talk about all kinds of resources that are available around the world. But a number of these tremendously useful resources are housed at the National Library of Medicine. And we do provide training on these tools as well. But let me just also show you a graph of overall the growth of databases over the years. And this is a tool from this is a graph from our friends at GeoSpiza, and I've provided the link there. You can see that later on. They've got a blog post associated with that. So you can see the NAR database issue every year publishes a collection of resources, and what they have graphed is the number of biological databases in the NAR database issue, which you can see has grown over the years as well. So there are lots and lots of software tools out there, but software tools are also very challenging for a couple of different reasons. As Todd Smith of GeoSpiza has said here, in the end, biological databases really do model biology and the principles of evolution. Perhaps it's apropos that new ones emerge through speciation while others go extinct. One of the challenges in this field is learning to use resources effectively, and those resources need to be maintained over the years. But sometimes they are also going away, and you have to stay on top of which ones are currently still available for your needs. And some of them are evolving. They're adding new tools, new data types, new features. So one of the big challenges in bioinformatics is actually staying current in this field. So at Openhelix, that's our goal, to help you find the right resources that you're interested in and keep current with those as well. So what I'm going to do is I'm going to introduce you over the next half hour or so to a lot of different tools. I'll provide a list of those later so you can go and check them out. You don't have to take wild and crazy notes as we're going through this. But I'm going to talk about several different types of tools that researchers need to access. Some of those we refer to as core tools. So those are tools, large databases, or sites where you'll have to access a lot of different species information or many different types of data, for example. So these are big, heavyweight tools largely run by a big project team, and they provide a lot of information that most researchers will need to access at some point in their career. There's another type of tool, though. We call those community tools. And these tools might be, for example, a species-specific group of tools. So, for example, the zebrafish database. If you're a zebrafish researcher, you should also be familiar with that. Even if the genome data is also available in some of these big core tools, the species databases often provide special tools and connections to other researchers in the field that are really important to know about. So community tools are also really important. Sometimes there are disease area tools. For example, we're often asked about cancer tools because cancer researchers have specific needs and want to focus on specific data types as well. So there are these types of community tools that might be species-related or disease-related or something like that. But there's also another class of tools that we like to provide information about. These are handy problem solvers. If you're performing research and you've got a protein of interest, for example, and you want to diagram the mutations that you find on this protein or the domains that you've seen, those kinds of handy little tools also will make your work easier as you are presenting in classroom situations or in papers or seminars, for example.